Question 5, a question on plant reproduction with the groundsel. A plant that colonises open ground, crevices and walls and paving between paving slabs. Here we've got a diagram of it. Hopefully you can see some of the things that you recognise there. And labels here, stigma and anther. Don't get these the wrong way around. I got these the wrong way around on a previous version of this video. Quite often they're the other way around. The anthers are higher than the stigmas. Anyway, anthers, male, produce pollen. Stigmas, female, where the pollen is going to go from another plant onto this. A. State whether the florets shown in the diagram, so this is the whole floret, are genetically identical or genetically different. Quite common to get this wrong. These are just parts of the plant. So part of the plant came from the seed. It's only the gametes, so the actual pollen itself, or the egg cell, that's going to be different. And the rest of the plant is just going to be made by mitosis, from the original seed. So this is going to be answer the question, are they genetically identical or genetically different? They are genetically identical. And why? Because this plant just grows normally like you grow, and you use mitosis to make more cells, including those of the plant. So growth by mitosis. So just like your finger will have been made by mitosis, this plant has. The only differences will be in the gametes themselves. One way shown in the diagram which the structure of a floret reduces the chance of self-pollination. So self-pollination is when the pollen from the male part of the plant, the anther, lands on the same plant. So the stigma of the same plant. And you can see here the stigmas are well out of the way of the anthers, and so the stigmas above the anthers, and so less likely to be self-pollinated. So stigma above anther, they're not at the same level. It's not enough there to say different heights. You need to sort of get which one's which. Explain the advantage of preventing self-pollination. Well, if you're using your own gametes to fertilise your own gametes, there will be some slight variation, some mixing up of the genetic information, but a lot less than in normal sexual reproduction. So there'll be, the term we usually use is less genetic variation with self-pollination. So prevent self-pollination to increase genetic variation or diversity. And why would you want to do this? Well, if you increase variation, then if there's a new disease or a change in weather conditions or something like that, then you will have some members of the species that will be more advantageous, and so they will be able to survive in this changing environment. So ability to survive a changing environment, or perhaps a new disease. Then in part B, we've got a sorry tale from the Oxford Botanic Garden in 1690 of an escaped plant. You thought plants can't escape, but there they go. And we've got a 2N plant, fertile, and a 4N plant. So we can get multiples of our normal, what you think is normal. So you are 2N with your gametes N, and we can double up or even triple up and so on. And then we get, that's the native plant there, and then we get this sterile plant, and then eventually a fertile plant. And if you look at the numbers, we've got kind of the cross between the 2 and the 4, which is 3 sterile, and then we've got 6. So those numbers will be important later on. And normal 2N plants, well, they can produce gametes, because you know in meiosis 1 we get separation of the homologous pair, so it turns into N, so that's the reduction division, and then 2 gametes come together again to make 2N. That's not possible with something that's 3N. So you've got 3 copies of the chromosome. If you try and divide those, well, you can't put 1.5 in each, so you've got a 2 and a 1, and then there's problems from there. So generally sterile. However, if you've got even multiples of that, so if you've got 2, 4 or 6, then you can still get pairing up of homologous chromosomes and reduction division and so on. So the numbers will add up, and that you're going to use that later on. The question asks for SS. So leaf cells of SS contain 20 chromosomes. How much have we got in the gamete, the endosperm nucleus and the petal? So SS contains 20, so that's this one. So these leaves contain 20, so our 2N is 20. Our gametes will be N, so there'll be half of that. And the endosperm, well, if you remember your plant reproduction, you've got the pollen tube and you've got the male nuclei in there. Well, one of them fuses with a female egg to make the 2N zygote, but the other one fuses with the two nuclei, polar nuclei, to make a triple version. So you get the male nuclei and 2 female nuclei to give 3N, and so that will be 30. And so here our gamete, so our normal petal like leaf would be 20, because that's our 2N. Our gamete would be half of that, because that would be N. And our endosperm nucleus, well, that's 3N, and it doesn't actually make it into the next plant as it works, it gets used up, but that will be 30. Part 2, badly answered really, and mainly because the not looking at the diagram and not relating what you know to the diagram. X, so bacteri, has 3 copies, and describe the change that has resulted in the formation of cambrenensis. So that is half the question, and explain why it is fertile. So we've got 2 jobs to do here. First thing is, tell us how X came to be C. That's the question. So how, I'm just going to use the initials here, how that became that. A lot of people talked before then about what happened with the original 2 plants, but that's what the question is asking. So if we look at this, how did that, this is the first bit of the question, how did that become that? That's the part of the story. A lot of people talking about this stuff up here. Okay, so we're going from 3N to something that's 6N. And what must have happened? Well, what must have happened is this DNA, or these chromosomes, must have doubled up but not divided. So they've doubled up, just like in normal mitosis or meiosis. They've doubled up, but then they haven't split. And so that's the only way that you can get an increase like that. So the answer is going to be the chromosome numbers doubled, so from 3N to 6N, or if you use the words triploid to hexaploid. And now they've got an even number of each chromosome. And you can suggest how that might have happened. Here the cell hasn't divided, and you can say no cytokinesis has happened. Or you could even suggest, if you know about non-disjunction and chromosomes essentially attaching to each other. So let's put those answers in. So chromosome number has doubled. So we're just using the diagram for that. So that's gone from 3n to 6n. And then how might that have happened? So e.g. cell did not divide, or something like no cytokinesis. That's a very hard answer, is that? Fortunately, there's more than 5 things we can say in this answer, and there's 5 marks available, so you might not have said that yet, still get full marks. Next thing to talk about is, why is this fertile? So why is this 6n plant fertile? So 3n plant is not fertile, 6n plant is fertile. And to be fertile, we need to produce gametes. So the question is just really going, how can this produce a gamete? So we're back into your existing knowledge, and most of this is suggesting this is AO1, which is just coming up with your knowledge. I think that's a bit tough to say it's AO1. I think it's at least AO2. So your knowledge of meiosis, that's what we're really talking about. And so are we going about, if we've got an even number of chromosomes, they can now pair up. In what process, what stage of the process, and what can be produced. And so once you start this, you'll get all the marks. So if we say now, for cambrenensis, we can undergo meiosis, and that's 1 mark. And we can say in prophase 1, which is another mark, we get homologous chromosomes pair up, or you get bivalence forming. And so, the end of this process, now that these can pair up, we can get that first division, and then we can get the second division, the separation of chromatids, and we can get gametes. So, gametes can now be produced. And so those are 4 marks, separate marks there. But once you start that, you go, oh, just tell me about meiosis. And the last little part, cambrenensis appeared independently in Edinburgh. There's just a small number of plants surrounded by some parental plants that they developed from, and these were on some derelict land. Why they became extinct. So we've only got 102 plants growing on an area, a very small area that is derelict, and they're surrounded by lots of normal plants. So you can imagine various things that might happen. These new plants might have been out-competed by the other plants and so disappeared. That might be a reason. So they could be out-competed by the other plants, and it says what they called parental plants. You could possibly suggest that they weren't able to breed with each other or breed with the they wouldn't be able to survive because they wouldn't be able to breed. But more likely, if you look at this as a derelict land, well, if we make it derelict means it's been left alone and it's just kind of bricks and rubble and stuff like that, wasteland. But they might build a nice set of houses on there, and that'll all disappear. And there's only 102 of them, so that might be the end of that. So loss of habitat because they want little cracks and crevices and things like that. So if it was made into a playground or houses or something like that, then they've lost the place that they live.